I just wanted to remind you that this meeting is being recorded, and if you don't want to be seen on the recording, turn your camera off. If you want to leave questions in the chat during Lee's presentation, then please do that, and we'll come back to it at the end. So listen, it's my real pleasure to introduce to you Lee Hickey from QAFE, Queensland University Agricultural, can't remember what the AFI stands for, but Lee will probably be known to most of you for the kind of pioneering work that he's publicized and developed beyond his probably what was capable of in the past, which was speed breeding. And almost everybody that works in an institute, certainly maybe even in a university, is using that approach to advance generation times to 4 or 5, maybe even 6 generations per year. That was a major development, I think, and it's democratization, I think, to the rest of the community has been really good. So I just want to welcome Lee. He's actually in Copenhagen just now, so I don't know what he's doing there, but maybe he'll tell us at the end. But he is enjoying some barley product, I'm told, maybe with some wheat in it as well. I'm not so sure about that. But he's going to talk about something which I've never heard him talk about before today, and that's about roots, machine learning, and genome editing. There's quite a lot of stuff going on in machine learning and artificial intelligence now in agriculture, and I'm interested to find out, as I said to him just before we started, if the talk lives up to the hype. I'm absolutely sure it will. So Lee, over to you, unlocking the hidden half.
Thanks a lot, Robbie, for the introduction, and it's a real pleasure to be able to share some of our research today as part of the International Barley Hub seminar series. So thanks very much. It's great to be here in Copenhagen as well, and actually I'll have the chance tomorrow to check out some of the root phenotyping facilities at the University of Copenhagen, which will be really interesting as well. So on that topic, I'd like to kick things off and share some of our research in this area, trying to bring together and utilize some of the new technologies. So I wanted to start with, I guess, the question around why are we even interested in roots? And I think when we think about the big picture, really what we're trying to do is improve the yield stability or consistency of barley production across seasons. And so in Australia, these statistics came from a survey of farmers recently, where on average a barley grower can make a profit of a quite considerable profit, but in the dry years when water is not sufficient, that completely flips around and becomes a loss in those dry years. So really improving the stability of the varieties that we grow on farm is important for the future, and even more so in the face of climate change, where these fluctuating environmental conditions are likely to continue. So when we think about roots, they are pretty important. They're underground; we can't see them, but this is how the crop is taking up water. And so, you can think about the distribution of roots underground if you have genetic variation for this trait. If you have a shallow root system, it might be advantageous for capturing in-season rainfall in shallow soils or in high rainfall zones where you're growing barley. But a deeper root system might be advantageous in deep soils where you're under terminal drought conditions, and therefore enable the crop to avoid drought stress and still fill plump grain for harvest. Of course, roots are also important for nutrient acquisition as well. And so if we layer this over in terms of where the nutrients might be, so most of our nutrients are in the upper soil layers, of course, but there's also opportunities to tap into leach nitrates as well. And also management practices now are considering this, trying to place the nutrient where the actual roots are. And I see this could be something in the future that we work towards, trying to bring together these optimal root systems and management practices on farm to maximize yield. When we think about modern barley breeding, it's had a huge impact in terms of improving productivity. So that's yield, quality for the malting industry, for our farmers around the world, really. Of course, that's come at a trade-off in terms of a reduction in genetic variation to achieve those genetic gains. But all in all, it's been hugely successful. The lack of genetic variation is problematic if we start to think about new traits that haven't been selected for potentially. However, these root traits could very well have already been indirectly selected through selection in modern breeding programs, because they apply selection for yield, for performance in those target environments, and therefore, in theory, the optimal root system in those environments would be underpinning performance and could actually already be captured in our modern varieties. The alternative scenario, of course, is that we've actually lost variation for root traits in our modern varieties. This could be the case for traits like root hairs, potentially for nutrient uptake and capture, could be the case where modern production systems have high inputs, particularly for fertilizer. And so, for some root traits, there could be advantages in going back and actually looking at some of these land races. Another trait could be root biomass, for example. But in a lot of our modern agricultural systems, I guess the current theory or hypothesis is that, yes, we need roots distributed where those resources are in the soil, but that root system also needs to be efficient. So we're not diverting a lot of energy below ground unnecessarily. So that energy could be used to produce yield, right? So why aren't barley breeders selecting for roots? Well, I have to say they face a lot of challenges on this front. Root traits are very, very difficult to work with, and I don't know how I started working on roots. I must have done something wrong in a prior life or something. But actually, I do find it quite challenging and interesting. So and really interesting to apply some of the new technologies too. So yeah. The main challenges, well, I'll get back to the story. So the first one is really around the difficulty in phenotyping these traits in the field. You've got to dig up the roots. Very low throughput, only can phenotype small numbers of lines thoroughly. This is problematic. A lot of researchers, including us, our team in Australia, have looked at, okay, well, can we target proxy traits in the glasshouse that might be correlated to the field? Unfortunately, when we look at the correlations, they're quite weak. And that's not surprising because root systems are very plastic, and measuring traits early in plant development may not necessarily be capturing the full complexity of the root system at the mature stage in the field as well. The second challenge for me is really around the value of the root traits themselves. A lot of these root traits, we require more data to enable breeders to actually make decisions about selection for which root traits might be advantageous or preferred in which environments. That information is very hard to generate for the first reason around phenotyping. And the third point is around generally we've got a poor knowledge of the genetic controls of root traits. Root architecture is very complex. It's very plastic. It's difficult to phenotype. So these are all contributors to the fact that we've got a relatively poor handle on the genetic controls in comparison to a lot of canopy and above ground developmental traits. And of course, this goes hand in hand with also the lack of large effect QTL or genes that breeders could more simply adopt and deploy in varieties as well. So a lot of these problems are interconnected. So I guess when we think about if breeders wanted to go down the path of modifying root systems in elite varieties, you could think about taking sort of two paths with the main technologies we have at the moment. So we could use genome engineering to create novel variation directly in our elite barley varieties, or we could try to harness this natural allelic variation we have either in the elite breeding pools, if that's sufficient, or go back to these land races in gene banks and try to bring in that new genetics into the elite breeding pools for root traits. So if we head down the first path of genetic engineering, I'd like to show some of the latest results that some of the PhD students in my team are generating. And one of the genes we've been working with is this CEPR1. It's a peptide hormone receptor. It plays an important role in general plant growth and development. And a lot of this work had been previously done in Medicago and Arabidopsis. And you can see some of the results on your screen where if you knock out this CEPR1, you get a much more narrower root system and deeper root system in these model species. In fact, there's a wealth of information around developmental genes in these model species that we can try to draw on and maybe take a shortcut in engineering these traits in our cereal crops. And so obviously the most important cereal crop is barley. And so in theory, if we can get directly to some of these genes in the pathway, then maybe this is a shortcut instead of trying to deal with the complexity of the genetics in barley as a species. So that's the whole idea. Can we leverage this prior information that we have on the model species? And so we set out to knock out this CPR1 gene in barley, and this is work done by our PhD student, Richard Dixon. If you follow the next few slides, you can see Richard's face move around different parts of the screen, which is fun. But essentially, we knocked it out in Golden Promise, and the first part of the story is we've measured root angle, and we find, just like the model species, we get a significant reduction in root angle in these knockout lines compared to Golden Promise. And when we talk about seminal root angle, we're measuring the first pair of seminal roots that emerge from the seed. So this is very, very early in development. And as I mentioned earlier, we know that this trait is not 100% correlated with root system architecture, mature root system architecture in the field. And so then the next step was for Richard. He wanted to know, okay, well, how does this the knockout of this gene affect the root system? And so he set up these homemade rhizo boxes. They were quite wide, so we didn't want to constrain the root system because if you have a narrow rhizo box, you kind of train the roots down. So he made these extra wide chambers up in our PC2 glasshouse, so we could actually measure it on these transgenic lines. And you can see here the narrow root system of the knockout line compared to Golden Promise. And when we do image analysis using RhizoVision Explorer, we can actually quantify the root angles. And overall, in the root system, we see a much higher proportion of steep angles in that edited line, which is the knockout line, which is pretty cool. When we look at the root distribution, so where are the roots in the layers of the soil, we can break up our images into sort of squares or a grid system like this. This type of analysis and setup was sort of inspired by the Manchadi paper on wheat quite a few years ago. We can look at, okay, well, the proportion of roots in those zones, and you can see really nicely that in the knockout line, we've got a lot more roots in that those squares 2, 5, and 8 going down in the middle, directly underneath the plant. However, you'll notice that the roots aren't significantly deeper. So in that bottom row of squares, we're not seeing more roots. It's a narrower, more compact root system, but they're not deeper. So that's something to think about in terms of this gene and pathway. We've got to really think about other genes that could be packaged with this gene potentially that actually enhance the elongation of the roots so they grow longer, if that's a desirable thing for some production systems. The efficiency and compactness of the root system is quite interesting as well and could be valuable in an agricultural system. The next thing Richard did was he looked at the shoot traits, because we often find that these root developmental genes are closely connected with the shoot. And unfortunately, with this gene, we did see a shoot penalty. And so here you can see we have a reduction in spike length, a reduction in grain weight, and so we're getting smaller grains per spike, and smaller spikes. So potentially not a good thing for high yielding environments if we're reducing the capacity of the plant to produce lots of grain and that are plump. So this is something that we're really interested in measuring in the field. And when it comes to these, like I said, these root developmental genes that we're looking at, this shoot trade-off is a common feature. And it's no surprise, the roots are connected to the shoot. And so we're exploring ways in which we can start to decouple these root and shoot traits. And one of the approaches is that we're taking is using a genome editing approach. So it's an SDN1 approach, or a knocking out components, not inserting. We're working with Dr. Peter Crisp, who specializes in epigenetics, and we're looking at editing some of the regulatory regions around these genes. And so some nice work in this area using this approach in tomato shows that you can create quantitative variation and even decouple some pleiotrophic or correlated traits, which is really interesting. So we're looking at promoter regions and cis-regulatory elements that we've identified as candidates that are nearby the gene of interest to try to break these relationships. So we'll see how we go on this front, but we've edited a number of the regulatory components already, and hopefully shaping up to phenotype those soon. The next gene I wanted to talk about was a PIN2, and PIN2 is a bit of a different gene. In Arabidopsis, it's an efflux carrier involved in polar auxin transport. And you can see there in the screen on the left, on the images on the left there, what happens when you knock it out. You basically, the plant loses its ability to respond to gravity. And it's really clear impact on the direction of root growth in Arabidopsis. But in rice, they found that overexpressing PIN2, they actually found some effects on canopy traits, which were really interesting. And so part of our research was really to explore, well, what is the function of PIN2 in barley, and is it root specific, or are we seeing shoot impacts as well? And so the UQ team and Zach knocked out this gene PIN2 in Golden Promise barley. And the first thing we did was we performed this gravitropism assay, where we plate up the seeds of barley, let them germinate, and then rotate them, and then measure the root's response to that response to gravity. And as you can see here, the PIN2 knockout lines don't respond to gravity at all. The root just continues to grow on its original trajectory despite being rotated. So there's an absolute lack of response, which is really cool to show this in barley. So that is maintained from the model species into barley. And I have to say that these genes aren't always consistent in terms of their function from model species to serial species. Often the complexity of the serial genome really changes the story and the function of the gene, and the pathway is often more complicated. So we were quite lucky from this perspective for this particular gene. The next step was to then look at the effects on the root system architecture, and Zach grew them in rhizoboxes, as I explained before. We have this grid system, and you can see the knockout line here in pink for PIN2. We're getting a lot more roots, particularly on the edges at the very top of the rhizobox, even filling the corners. So we're getting a wider root system, a shallower root system, as a result of the roots not going downwards, essentially. They're just spreading out all over the place. And generally speaking, they're occupying more of the upper layers of the soil, which is really interesting. So maybe this gene could have a benefit in higher rainfall environments where we do want the roots in the upper soil layers. And so the next step, Richard, as part of his PhD program, he's going to be testing some of these edited lines, so the PIN2 knockouts, the SEPR1 knockouts, along with the Golden Promise wild type. And he's going to test these in the field in collaboration with the University of Adelaide, And you can see there a picture of Stuart Roy, where they've got a nice GM field testing facility down there. So hopefully we can, validate the effects of these knockouts on roots, but also the shoot, and measure yield as a result. So it's really nice to see this work transition from a glass house or lab initially to a glass house, and now finally to the field. So we're really excited to see them growing in the field where they belong. So the next part of my presentation was really to highlight some of the work we're doing around phenotyping root systems in the field. And as I mentioned, this is a real bottleneck in trying to get to the genetics and for breeders to work with these traits. And we know UAVs or even satellites now present, really exciting opportunities for phenotyping traits at scale that we need for plant breeding. And when we think about some of the sensors that we can load on our UAVs, they can measure traits more accurately than the breeder's eye, but they can also detect traits and features of a crop that the human eye cannot see. And so here you can see this is a field that we're flowing with our multi-spec sensor. This is an RGB mode at the moment, so this is what the human eye sees. But when I click my mouse, I'm going to switch to thermal, and this will reveal the strip down the bottom, which was hidden below on the RGB mode. Now we can see where the irrigated strip is. That's highly transpiring. So the crop has got more access to water in the soil, and so this is reflected in the temperature of the canopy. So just a good example of these sensors can actually capture things that the breeder's eye can't actually measure as well. And so if we think about roots, could these sensors actually be picking up signatures in the canopy that are perhaps related to where the roots are in the soil profile? And so our hypothesis here is really on your screen. So it's the chemical composition of the canopy at key growth stages captured by multispectral imaging can reflect genetic differences in root distribution, supporting the use of UAV-derived vegetation indices to indirectly predict root traits. And this is where machine learning comes in, because we can actually build the models using these imagery data to predict the root system. So that's the whole idea. So to explain some of the work that we're doing, I wanted to introduce the germplasm. So this is part of a collaboration with Intergrain. We're working with a panel of about 400 spring barleys, which are really representing genetic diversity of the modern breeding germplasm that Intergrain work with for breeding in Australia. It's genotyped with 6,700 XT SNP markers, which is what they use, the platform they use for their breeding. You can see there the population in terms of the diversity. To test this hypothesis, we set up a field experiment where we had a couple of experiments in the same paddock. So in blue, we have what we call a root coring trial, where we've actually dug up the roots of 20 barley genotypes. And we've used a RCB design with 6 reps of those 6 plot reps of those 20 genotypes. Next door, we have the main trial, where we have that panel of barley breeding germplasm of about 400 genotypes in a P-rep design. And we flew our UAV across this entire site weekly from the time of sowing all the way through to harvest. And the idea was to use the root data that we dug up as a training data set, coupled with our UAV data set, to predict the roots onto our main trial. And an important thing to note here is that the composition of the 20 genotypes is very diverse. It's capturing about 97% of the allelic variation in that main panel. So we've actually used our core hunter to pick out that subset of lines in the coring trial. That's really important. Okay, so to explain our UAV data set, we've captured, like I said, flights weekly all the way from sowing through to maturity. So this gave us about 17 UAV flights. I think we had to discard a couple because they were poor quality. we ended up with about, 65 vegetation indices extracted for each of those 17 UAV flights. And essentially that's our UAV data that we're feeding into our machine learning model. We've also sampled canopy biomass at key time points, so tillering, stem elongation, and flowering. And we're using this physical canopy biomass data to also build prediction models using the UAV data to predict biomass at those key growth stages as well as roots. So digging up the roots, the hard work. Well, we use this technique, which is quite efficient, called the core break method, developed by Anton Wasson at CSIRO. And what we did is we took 4 cores per plot, 2 intra and 2 inter row, down to about 170 centimeters. Once you got beyond about 150 centimeters, there was very low genetic variation and the variability was very high in terms of roots. What happens is when the core comes out, it gets broken up into 10 centimeter sections. And when you break that core section, you count the number of roots that you see. So that's a pretty efficient way to get a root count data down the soil profile. Anton actually has a nice blue box that he's developed as well, which actually images these cores as they come out using blue light. So the white roots really light up in that image-based system. And in fact, that system's even better because it removes the human error associated with counting roots. And so we washed out a proportion of these roots as well to show that we're getting a nice correlation between actual physical root biomass and these root count data set that we generated. The modeling of this root data was rather complex because we're dealing with a spatial design, but also the roots vary downwards. So we adopted this longitudinal modeling approach from the Wageningen team to do this using a spline approach. And here you can see basically where we've got those root distributions for our 20 genotypes going down from a depth of 0 to 170 centimeters down the profile. And we can think about, extracting different information from these curves. We can extract area under the entire curve, which would be a proxy for root system size, or we can actually pull out the root counts at these specific intervals. And these reflect the different soil types, these soil horizons for the soil type that we're working with. And that's exactly what we did. So we're working with 5 root traits, each trait in each horizon, plus the whole area under the curve. So just to visualize some of these effects for the 20 barley varieties that we physically cored, here's the output from the modeling work. And we can plot these root counts as a variation from the trial mean. So you're looking at plus and minus root counts for these different varieties. And this is really exciting because it's really the first time that we've physically measured root distribution in the field for Australian barley. And we've revealed that there is quite significant variation among our barley varieties, which was really promising to see. So even varieties like this, you can see quite contrasting rooting patterns where, Spartacus has more roots on average in the upper layers, less roots in the upper layers and more roots deep in the deeper soil layers compared to this breeding line from Intergrain, for example. So really promising to see. The next step was to then use that physical root count data in a machine learning model to then, in combination with our UAV vegetation indices, to try to predict the root traits on the broader breeding panel of about 400 barley lines. And so we tested a partial least square regression approach and a random forest approach. Overall, I'd say that they were quite similar in terms of accuracy. However, the partial least square regression approach we went with this one, and it seems to be consistent across seasons as well. Now we've actually done this for a second season, and it actually turned out to be a drier conditions. And this prediction approach worked even better because the drier conditions were enabling us to better differentiate the root distributions among the barley varieties, which was pretty cool. But overall, getting some nice accuracies in terms of our ability in the test set here to predict roots onto the broader panel. So now we're dealing with predicted root traits using this machine learning approach on this panel of 399 barley lines. You can see the 5 root traits down the bottom here, area under the curve, 0 to 20, 20 to 60, 60 to 100, and 100 to 170. That should be 170. Okay, so this is pretty cool. We've got predicted root traits, but how is this working? So when we had a closer look at, so what are the vegetation indices that are contributing to our root predictions? And here you can see a bit of a visualization of that, the top contributing vegetation indices for each of the root traits. Overall, some key findings here are that actually vegetation indices captured across the growth cycle were important predictors of some root traits. And you can see there even some of the vegetation indices captured at just 11 days after sowing were useful in the root prediction, for example. So I think this is pretty cool because we're actually using the vegetation indices across the entire growth cycle, whereas we've got root trait data captured at one single time point, which was flowering. So we're using all that information to predict roots at that critical flowering stage. Key vegetation indices also appeared to be NDRE, ASAVI, and MSAVI, which is interesting. And measures captured just prior to flowering and during the early mid-grain fill period were consistently important. And that was also the case in our second year of data as well, which was really interesting. And that makes sense, I suppose, because that's when the crop is filling grain, the distribution of roots is pretty much set, and so we might be seeing signatures in the canopy, associated with where the resources are in the soil and where the roots are. So one thing that was really promising to also see was the heritability of these predicted root traits being quite high. So here you can see really moderate to high heritabilities for each of the root traits, capturing a high degree of genetic variance, which was really, really promising. The next step was, well, can we use these predicted root traits now to get to the genetics? And so we use this haplotype-based mapping approach. I guess it's a bit of an alternative approach to QTL mapping and has some advantages, particularly when you're working with breeding materials, when you're interested in tracking haploblocks or haplotypes through breeding materials rather than single markers. Just to give you an overview of the approach, I can explain it very quickly. So first, we run our genomic prediction model, backsolve those SNP effects. The second step is then breaking up each of the chromosome into chunks. So based on LD, these are our LD blocks. We then sum up the SNPs within each block, and this gives us our haplotype effects. So the different marker configurations within a block are essentially haplotypes. And we call this our local GBV because the traditional approach to a GBV is actually to sum up the SNPs across the entire genome, whereas we're doing this at a really localized region within a haploblock. We can look at a block variance, which is the variance in haplotype effects at a particular region, as a way to indicate whether this is an important block for our trait of interest or not. So this is the approach we used, and here's the results. So we've done the haplotype mapping for both the shoot traits and the root traits that I mentioned. So we had the predicted canopy biomass at the 3 growth stages, and we had the predicted root traits, and there were 5 of those, if you remember. So the inner rings represent the root traits, and the outer 3 rings represent the results from the canopy biomass. And so in short, what you can see here is in green, these green dots you can see are the top haploblocks. So the most important haploblocks are controlling or associated with canopy biomass, so the shoot traits only. The purple dots are the haploblocks that are most important for root and shoot traits. So they appeared in both analyses. And interestingly, the brownish dots are the haploblocks only highly important for root traits. So you can see, while there's commonality between haploblocks for root and shoot development, which makes sense because we know there's master regulators that are controlling above and below ground development in our plants. But there are, interestingly, blocks really important for modulating root traits only, which I think opens the door to some, interesting opportunities around modifying root architecture while we try to maintain the appropriate flowering windows and shoot development that we need in these different environment types. So the question we wanted to then answer was, well, are we seeing artifacts? Are we mapping, root genes, or are these things real, basically? So what we did is we overlaid a lot of the QTL information for root traits that from the literature, plus some of the known genes controlling root development in barley. And so here on your screen, you can just see an example for chromosome 5H, where we've actually positioned EGT2. It's a gravitropism gene affecting root development in barley. It was recently cloned, as well as VERN1 in barley. And we know this VERN1 gene also has a role in developing root architecture below ground as well as above ground for the flowering pathway. And so we found some really nice alignment with our root blocks with some of these key regulators of root development, which was really nice to see. So in terms of the application of these results in a modern breeding program for barley, well, how would we go about doing that? So here's a slide providing one way to do this through haplotype-based breeding approaches. And so these are some ideas that we've put together from with our work with Mark Cooper and Owen Powell. And so it's a nice summary of this. So essentially, if we can phenotype our breeding populations, or we can engineer new trait variation and we introduce this into the breeding program, and we get to some of the haplotypes that are important, that are controlling these traits, we can take a more targeted breeding approach and direct genetic gain for those traits underpinning performance in the different target environments that we're pursuing. So I think that's generally what we're trying to do, and we see a role for these key developmental traits or traits that underpin performance in these target environments. I think we've still got a long way to validate which of these root traits are important in which of the environments. That's a really big gap that we've got to answer before we go down this path. So on that note, I'd like to finish on some future directions and opportunities. So from the first section of my talk, genome editing, we can edit key genes in the developmental pathway for roots, and this actually leads to some pretty dramatic modification of the root traits. And I think that's pretty cool because we can leverage or learn from these advances from the model species. But sometimes their genes don't translate from model species, but sometimes they do. And when they do, they have a big effect, which is pretty cool. However, we've really got to focus on editing elite barley varieties, not Golden Promise. We've got to progress from Golden Promise, make sure we're editing these targets in elite varieties, because then what we're generating is actually useful for barley breeders. I think it's also important for us to think about ways which we overcome the shoot trade-offs as well with these developmental genes we're trying to manipulate. I talked about engineering some of the regulatory elements, for example. Fine-tuning the trait expression is also important because, with root traits, like most physiological traits, the relationship with yield is not linear. And so we often have a sweet spot that we're aiming to achieve for a particular production scenario. So how do we do that with a technology like gene editing? For the second part of my talk, I think there's a real opportunity here to scale phenotyping for roots, but also other traits using this type of approach. Building training data sets to extrapolate complex, difficult to measure phenotypes across breeding populations at scale is really what we need if we're going to be considering these traits for selection or using them to get to the underlying genetics. And I guess some of the key questions is around advancing this technology further. Well, we've looked at 2 seasons already using the same soil type. When we do the haplotype mapping on that second season, what we find is some overlapping haploblocks, but largely speaking, quite different genetic controls. I think this highlights the high degree of plasticity we have when it comes to root traits to deal with in terms of water availability across seasons. That's using the same soil type. A big question, I think, going forward is this approach also applicable to different soil types in environments? Can we get it working in shallow, sandy soils? That's yet to be answered, hopefully, using a similar approach. But I think, again, this sort of data will be very valuable for us to answer that critical question about trait value before we can actually start to select for these traits in a breeding program. And I'd like to acknowledge our funding body, so mainly the ARC and our partners, Intergrain. Of course, the large team of people at UQ around the various aspects of what I presented, our collaborators at ANU as well on some of the gene editing work, translating the model species genes into barley, and of course, our Anton Wasson at CSIRO. Thank you very much.
Lee, thank you. Fabulous. Well done. You got through from the beginning to the end. So before I ask people if they've got any comments to make, I wanted to just say something. It's amazing how Owen Powell closely looks like his dad. So for those of you on the call who don't know, Owen's father is a guy called Wayne Powell, who we've worked with here for a long time. Very good barley geneticist, currently the CEO of SRUC in Edinburgh. And
we're calling him an honorary Australian now because he just bought his own home in Australia, and he's great to have around at UQ. Really smart guy.
All right. So does anybody have any questions from the audience? I can't see hands going up, by the way. So if you want to just start speaking, that would be very good. And there's lots of people who work on roots online, so I think you should be having some questions. Can I see any questions here?
Oh, there's a question here in the chat.
Do you think the sample size? Yeah,
sample size is enough for building a machine learning model? It's a good question. I mean, more the better. We were getting quite nice accuracies for the canopy biomass predictions and the root traits using that data set size. But I think that comes with 2 sort of considerations that were important. One was the diversity in the training set that you're working with. So if you're dealing with something, if you're trying to put into a population that's more diverse, that's where the model's going to fall over and not perform very well. So it was really important to make sure we had a really diverse subset. It's only 20 genotypes, but I think we had 6 reps. So we talking about 120 plots that we're generating data for to then train and predict across the larger population, which was about 600 plots. Yeah.
Malcolm, I can't see anybody's hands up, and I'm not sure if you can. So in the meantime, I'm going to ask a question. Lee, you seem to focus quite a lot on using the information that is coming from model species as a predictor for what's going to happen in cereals. Have you thought about using any other type of data from cereals that might lead you to discover things which have not been discovered in the model species. So I'm thinking of things like a lot of the very deep transcriptomic data sets that are available, where you get specific expression of transcription factors in root tissues which are not expressed in other tissues. Have you thought about doing that?
Yeah, that's a good idea. No, we haven't done that, but that's a great idea. I think there's probably also opportunities for machine learning to, you know, to be connecting data sets from studies that have been done too, from that perspective. So
Yeah,
can we, yeah, connect and leverage those data sets to identify targets? I think could be pretty cool.
yeah,
That's one of the challenges with all those prior studies. It's often they're genotyped with different marker platforms. Markers are aligned to different versions of reference genomes.
of course.
So
Yeah.
to integrate sort of QTL studies, it usually takes a PhD student half their life to position everything. But maybe with these new tools coming online, we can do those sorts of tasks much more efficiently. Yeah.
I think there's opportunities there. In fact, I know there's a fairly big effort to use that type of approach to start to dissect root traits in different organizations across Europe. Abdul Aziz has a question. What's the spatial resolution used on the UAV for the imaging for roots?
Good question. I don't know that off the top of my head. So we fly our drone at, I think, 40 meters. So anyone who's familiar with the sensor will be able to tell you what the pixels per square meter is from that distance. But I don't know off the top of my head. I'm sorry. It's not high resolution for sure. You could think about using hyperspectral imagery too to try to take this approach to the next level as well. Maybe that helps improve your predictions.
You mean ground held, or
Oh,
do you mean
sorry, Robbie?
ground held stuff? So high resolution from cameras held on poles.
Yeah, you could try that as well. I think I hope this is just the beginning for this type of approach because I think we need solutions to scale basically, and um, actually everything I presented today, we actually haven't published at all. So uh, hopefully it was interesting from that perspective, and we're sharing some of the latest work. But we just wanted to, for that root prediction work, we just want to get the idea out there, and so people can start to try this themselves and take it to the next level as well. It's just the beginning, I think, in how we can go forward. Hopefully it evolves. Yeah.
Okay. So Gwen has a question. Do you want to ask the question yourself, Gwen? I can't see, so I'm going to ask it for you. So Gwen Kirsten has a question, which it said, great presentation. I think we all agree that anyway. So regarding the PIN2 and CEP1 receptors, did you hit the right gene in the first attempt? And was that the right gene only based on similar protein sequences or not?
Yeah, that's a good question. I don't know if I can fully answer that. There are a number of orthologs or similar with high degree of similarity in the barley genome when we look at the Arabidopsis and Metacargo versions of those genes. I know, there were quite a few that I think that Karen and the team, took a few different approaches to try to narrow the targets down. Not to say that some of those other genes might also be important, I guess, too. I think it's interesting to keep in mind. Translating this pathway or getting it to work in like a crop like wheat too would be probably even more challenging with with 3 different genomes and maybe multiple targets that are quite similar. So it's always a tricky thing. I'm sorry I don't have all that information in my mind right now, but I can get back to you if you like.
Yeah, I thought Gwen was going to ask something about the EGT gene, but obviously not. So SimSip has a question as well. He again says, fantastic talk. Again, we all agree. How do you approach integrating multiple UAV flight time points into the models? I had exactly the same question.
Yeah, well, we're using the same pipeline, so that the images, they get stitched for each flight. We then have a grid that pulls out the indices for each of the plots across the trial, so every flight is sort of strictly adhered to, protocol wise, to make sure that the data is very consistent. There's a lot of checks that are put in place. A lot of the commercial providers have very similar checks and setups and that sort of thing. That's critical to make sure that your flights are integratable. Essentially, though, we're working with those checks in place, and we're then working with the derived vegetation indices at the plot level. That's the input into the model training. So that's a good question. There's a lot of challenges around integration of flights at different time points and even across environments as well. So you make a good point. And I think one of my postdocs is actually really interested in this question, which obviously a lot of other people are interested in too. But what I was going to add was that you mentioned the gravitropism gene, Robbie. And I think that's really cool work as well. And I think in that case, the knockout actually leads to a narrower root system as well, which is quite handy. But on reflection of this work in barley, if you think about the gravitropism genes, the PIN, the SEPR, I mean, we're getting quite big changes in root architecture as a result of knocking out a single gene, which is pretty exciting, because when we look at effects of QTLs and we map them for these traits, we generally have much smaller effects. And I guess, that could be a reflection of directly knocking out a key component in the pathway that's maybe highly conserved in barley potentially. I'm not too sure, but I think that's something I've certainly observed.
So along the same lines, did you, having looked at some of these genes where you've got knockouts and getting fairly significant effects, did you go back to the natural variation and survey the amount of genotypic variation there was in that gene in a natural environment or a natural collection of germplasm, maybe across a wide geographic or ecological range?
Yeah, that's a good question. I think Zach and Richard have both done that for both of their genes, looking at basically the haplotype variation as a starting point in sort of gene bank materials, and I think that revealed pretty low variation or very minimal or no variation. And then I think they looked at then sequencing the gene in sort of representatives or clades of diversity, essentially. And they found some variation, but I think it was in pretty much the sort of non sort of functional parts of the gene when they sequenced the gene in those representative materials. So from that perspective, it was, I think, that these two genes at least pretty conserved in modern barley. And so what we're seeing is a result of knocking out these things for the first time, I suppose. We haven't sequenced the gene in every barley, that's for sure. So there could be natural variation out there. And I guess that was the idea, was could we actually find natural versions of these genes and avoid, CRISPR applications as well, where, some regions it's not an accepted technology, unfortunately. Yeah.
Induced mutations are fantastic though sometimes, because they do give you quite extreme phenotypes. There's one last question I'm going to take before we wrap this up, because time is moving towards 2 o'clock. Doesn't this introduce data leakage during the training and validation? Maybe Hollander wants to expand on that a little bit, if he wants to unmute or she wants to unmute. I'm just reading the question. Hollander, do you want to?
Just give me a second. I'll make sure that they can unmute.
Essentially, we're treating the biomass predictions at the different growth stages I referred to. I mean, we're just considering them to be different traits in this scenario. You can do a lot more with that data. Of course, you can actually model and predict biomass over time in terms of the dynamics of the growth cycle as well. But we took a rather simple approach or view for the traits for this study.
Brilliant. I hope that answered the question.
Probably
Lee,
not.
one
Probably not.
Yeah, one comment I just wanted to raise, and you probably know the answer to this anyway, but I believe the people in the plant accelerator in Adelaide have been doing quite a lot of this, measuring traits over various time periods and working on splines. So, to actually extract some of the I guess the underlying biology behind some of the measurements that they make. And some of the statisticians there may already have some methods for taking that type of data and then quantifying it or analyzing it over time. So just a comment. It might be useful to reach out to some of the people. Stuart Roy should know them.
Yeah, absolutely. Yeah, no, I think it's I think it's sort of good to try to think of solutions for this for these root traits, which are so problematic. I mean, I've sort of felt a bit frustrated working a lot on glasshouse root traits in the past. Initially, in the early part of my career, and it turned out they weren't very useful. And some of these traits you can measure early into plant development have turned out to be quite useful for other crops like sorghum. They've exploited that quite well. But for wheat and barley, it just wasn't. It's just not the same. I guess it's not surprising in hindsight that these root traits are so plastic, and you've really got to measure them kind of in the field. But that brings its own challenges. So I think hopefully it's just a step in the right direction anyway. Yeah.
Brilliant, thank you. Listen, it's 3:00 on the button. I just want to thank you again, Lee, for taking the time to give us a presentation about some of the stuff that you're doing, which, like I say, I'd never heard about before, which I'm really pleased to hear about. And I'm sure everybody will join me in thanking you for such a great seminar. So thank you so much. Enjoy your time in Copenhagen. Say hello to Owen Powell from me and from Joanne Russell and others that are potentially online. It's a long time since I've seen them. So thank you.
Thanks
well
very much.
done. Thank you very much. Great
See
to hear.
everyone.
Take care.